Hi, buongiorno. I'm going to be speaking in English, so I'll try and speak slowly for the translations. As mentioned, my name's Kristy, and I'm currently conducting my PhD under the supervision of Professor Joseph Getz, who you've all heard about, and Dr. Ewan Farm, and they'll be both presenting a little later today. Our lab is actually located in the building pictured on the slide. That's the Adelaide Health and Medical Sciences building in Adelaide, Australia, which is where I'm from. So I'm really grateful to have been given this opportunity to talk to you today. I thought I'd just briefly start by telling you a little bit about myself. I completed my undergraduate degree in psychology at Macquarie University in Sydney, Australia. After that, I spent a year conducting research in the field of neuropharmacology before making the move to Adelaide to join Joseph's lab. Now, I've really enjoyed this opportunity that Joseph has given me. He is a great person to work with. Now, one aspect of my PhD, as mentioned,
Studio dottorale, come già stato detto, è stato proprio quello di condurre una revisione sistematica nonché un'analisi del PCDH19 e della letteratura che attualmente.
Now, a systematic review, just in case you don't know, is essentially a collection and an analysis of research studies using predetermined selection criteria. The criteria I used were as follows. The study needed to report the exact PCDH19 mutation. So that is the type of mutation, as well as where in the gene the mutation was located. Secondly, the study
Poi questo studio è stato pubblicato in inglese, ma
a process of evaluation by the scientific community to ensure that publications meet certain standards. Lastly, I only included original cases. In other words, if an individual was reported on more than one occasion, I only recorded them once. In addition, I conducted a meta-analysis, which resulted in some secondary criteria whereby a number of publications were selected for further evaluation if they also included relevant clinical data. So this data might be things like the age at seizure onset or the degree of intellectual disability. When looking at the primary inclusion criteria that I applied, this resulted in the identification of 38 different articles and 271 individuals, 12 of whom were male. Now, the review obviously encompassed quite a lot of data collection and analysis, and I'm only going to go through a subset of the results with you today. And the first thing I wanted to talk to you about was the cognitive profile. So this is essentially just looking at the degree of cognitive function or dysfunction and what proportion of individuals fell in any given category. So the review identified that there were 28% of individuals with normal cognitive function and 5% with borderline functioning. In terms of intellectual disability, 27% of patients were reported as having mild intellectual disability, 22% with moderate, and 17% with severe or profound intellectual disability. Now, looking at this on the whole, that's around 1/3 of individuals reported as having no intellectual disability and approximately 2/3 with some degree of intellectual disability. Now, it's important to note that these findings, even though I mentioned the review identified 271 individuals, these results are only based on 195 individuals. This is because not all reports contain information of this nature. And this is also important to note that the findings are based on what has been reported in the literature, so it might not necessarily be a true reflection of what's really going on. And this is something that we hope to address through my PhD. How we do that, I'll have a chat to you about in a few slides' time. If we look at the same data, but now also incorporating whether or not an individual also had behavioural problems, we can see that only a small proportion of individuals with no intellectual disability had behavioural problems or were reported to have behavioural problems, whereas a much larger proportion of individuals were reported as having behavioural problems in conjunction with intellectual disability. Again, we see a similar trend to the previous slide, with around 2/3 of individuals reported as having behavioural problems. Now, although, and I know it has been discussed, but although autistic, aggressive and hyperactive features were reported as the most frequent problems, information in this area is actually quite limited. So it's hard to say with confidence exactly what problems we're seeing and the nature of the problems and what proportion of individuals have any given problem. In terms of the review, however, I became particularly interested in this group of individuals, this particular subset, particularly those that had no behavioural problems. I wanted to know what was unique about this group. Why were they less severely affected than others? Could it be the type of mutation that they had? And it's not uncommon in the epilepsies where individuals with identical mutations have a very similar clinical outcome. So identifying patients with exactly the same mutation can be really beneficial, as it provides some scope for determining if any genotype-phenotype associations exist. In other words, whether the type of mutation is associated with clinical outcome. In terms of PCDH19, one such mutation has been identified, and that's the 1019A-G, which results in an asparagine amino acid being replaced by a serine at position 340. This particular mutation has been identified in 25 individuals, 20 of which are unrelated. If there was a genotype-phenotype association, we would see quite a similar clinical profile for these 25 individuals. I've listed 15 of them on the slide just as an example. As you can see though, the clinical profile of these individuals is quite varied, with around 50% of them having behavioural problems and quite variation in terms of cognitive function and dysfunction. Therefore, not really supportive of any sort of genotype-phenotype association in terms of the type of mutation. What was interesting that we found in the review, and there's a case listed on this slide, is that there are a number of females that have been reported as having a PCDH19 mutation and never having sustained a seizure, nor have they been reported to have any behavioural or cognitive deficits. We're quite interested in these females as they offer some scope for understanding what factors might be protective and how we might use this knowledge to better inform prognosis, treatment and genetic counselling. So hopefully we can identify more of these females so that we can conduct more detailed analysis. Now, I mentioned that I also did a meta-analysis, which essentially takes the information from a systematic review and looks at it in a bit more detail. Specifically, I was interested in whether clinical outcome was associated with a number of factors. The factors that I looked at were the age at seizure onset, so what age was the child when they first had a seizure, the mode of inheritance, so whether or not the mutation was inherited or occurred within the individual, or in other words, a de novo mutation, the type of mutation, which I've touched on briefly, or the region of the protein affected by the mutation. Now, of these 4 factors that we examined, only age at seizure onset emerged as being associated with clinical outcome. Specifically, an earlier seizure onset was associated with more severe intellectual disability than a later seizure onset, and this association was significant. This is the first finding to emerge of a potential phenotype-genotype association and has important implications for improved prognosis. However, to better understand these associations and to identify if others exist, we need to know more about these different clinical outcomes. We need to know more about unaffected males and affected males, as well as affected and unaffected females. It's important to get a clear picture of what's going on. So we know that from the literature, behavioural and cognitive problems are frequent, but we don't know the exact nature of these problems and exactly what proportion of individuals have any given problem. To address this, I have developed an online survey that comprises measures that are designed to address some of these requirements. Slowly? Okay. The survey has been developed in English as well as in Italian. It's been developed in a very user-friendly format so that it only requires, generally, the click of a mouse button to respond to questions or make selections. The survey takes approximately 45 minutes to complete. However, progress can be saved and the survey can be returned to at a later time. The survey is open to all individuals who have a PCDH19 mutation, as well as those who know someone who has a PCDH19 mutation. This could be parents responding on behalf of their child, or a friend or relative responding on behalf of an individual. This could be a male who is a carrier of a PCDH19 mutation, or a female who is a carrier of a PCDH19 mutation. We're keen to hear from both affected and unaffected individuals so as to get a complete understanding of the clinical profile associated with PCDH19 mutations. If anyone is interested in getting more information about our PCDH19 survey or how they might be involved, I encourage you to take one of the flyers that I've placed at the back of the room, or please come and talk to me in one of the breaks or at the end of the session. I'd be more than happy to answer any questions that you have. And I'd just like to conclude by thanking all the people that have been involved in this project, and especially to those who have already completed our survey, and to everyone today for listening. Thank you.
Thank you very much.
I'll ask you right face to face. So is there any evidence for gain of function of any of the mutations? And why can't you just argue that it's the variability, say, for a given mutation is just the degree of random X inactivation in a given individual?
Okay. So I'll comment on the X inactivation component because that's probably something that I've got a little bit more knowledge around, because that's also one aspect of my project. The degree of X inactivation being associated with the variability in the symptoms that we see is quite possibly a factor that is associated, but one that's yet to be determined, and actually forms part of my PhD, looking at whether different patterns of X chromosome inactivation are associated with the severity or the penetrance of the disorder. So whether or not those females that I mentioned who are unaffected, for instance, have a skewed X inactivation such that 100% skewing might then lead to a much milder or a non-existent phenotype, as opposed to what would be expected, which is a random X inactivation, which one would predict would be associated with the presentation of the clinical symptoms that we see with PCDH19 mutations. On the gain of function, I'm probably not the best person to answer that question, so maybe if there's someone else that wants to comment on that.
Stavolta funziona.
Yep. Thank you, Jack. Also for the comment about the lab picture. The gain of function, it's an intriguing possibility. So for those who don't really understand perhaps the gain of function model, it means that the variant PCDH19 would be destroying also the function of the normal PCDH19. So it's kind of dominant negative effect of gain of function which interferes with normal function of even the good protocadherin 19. This is potentially a possibility, and we're exploring it especially in some males, because there are some males where we see PCDH19 DNA variation, and we see that these males do have certain clinical presentations which may be similar or different to what we know about PCDH19. But these are males, of course. There is no other protocadherin 19 because they have only one X chromosome. But there is a possibility that if there are variants in certain part of the protocadherin 19, they may behave in a dominant negative effect. That's a possibility. However, I believe that in the PCDH19 epilepsy as we know it, and we generally discussing it, we probably talking about loss of function of PCDH19 and unlikely a dominant negative effect of gain of function, because we know, and people here in the audience found individuals with complete loss of one allele, and these individuals have very consistent clinical presentations when compared to the others, as far as we can talk about consistency in such a complicated disorder. So I would say likely not a gain of function or dominant negative effect in these individuals, but I wouldn't close the door on it, because we may not have seen all clinical phenotypes associated with PCDH19 as yet. I'll pass it on to Paul, and then Eric want to comment.
A very quick comment, Jack. We've looked at using a cell aggregation assay at a number of different mutations, including the N340S, and there's, at least in our hands, in those assays, they all appear to be loss of function.
Yes, I think that at the molecular level, we can say that the vast majority of mutations are loss of function mutations. But if we consider the cellular interference model, at the cellular level, we can say that we have a gain of function.
Do you want to comment something? In your experience and looking into the literature, which can be the strongest, because the incidence of cognitive, the intellectual disability and behavioral disturbances related with the presence or not of intellectual disability is the result of the description at follow-up, okay? In your opinion, not considering genetic differences, which can be the predictors, early predictors of the risk for intellectual disability, behavioral problems, intellectual disability with behavioral problems?
So what you're asking is what predictors might we use to determine severity of intellectual disability and presence of behavioural problems?
Yes.
Yes. That's a really important question, and that's one that I'm trying to answer. I think so far the discovery that early seizures being associated with more severe intellectual disability is a start to being able to answer that question. Obviously that's not something we can predict per se, but it's something that we can potentially try and prolong if we know that a child has a PCDH19 mutation, and we know that fever is something that generally is associated with the first seizure. If we can maybe prevent high temperatures or fevers and stretch out the point to which the child has the first seizure, therefore might then reduce the severity of the clinical outcome. I think that it is intuitive to some extent that having a seizure when you are very young, particularly during times of critical brain development, that this might go some way at explaining why early seizures are associated with the more severe clinical outcome. But I think that we definitely need to look at a much larger proportion of individuals to be able to better understand what predictors might be present.
Ci sono altre domande?
Hi. I have a question on the survey. It will be interesting, in my opinion. I don't know if you just didn't show it to us today. It will be interesting to add to that survey a time variable. I mean, how do the severity of the cognitive and behavioral disturbs change during time, during the age of the girls? I mean, from 5 years to 10 to 15 to 20 years old, maybe this level of impairment will change. So it's a very good idea. I like it. I'm very glad to contribute if I can, but this is just one suggestion that you might consider.
It's actually a very good suggestion, and it's actually something that has been embedded into the survey that I mentioned. We won't be able to look within an individual across time, but by looking at lots of different individuals at different ages, we can see if patterns emerge, like you've mentioned, whereby cognitive dysfunction worsens over time, or plateaus at some point, or improves. So yes, through the survey, we will be able to have a look at different ages and see whether or not there are patterns that emerge in that sense. So whether behavioural problems, cognitive deficits worsen over time, and to what extent.